Good morning, everyone. I welcome you all this national webinar on Lumpy Skin Disease, and this is being hosted by IIT Delhi and organized by our RCI partner SBB Tirupati RCI. Now I request Professor Reddy to please officially start. Professor Reddy, please.
Good morning, everyone. I'm audible.
Sure. Sir, your sound is a little low. Can we?
It's okay now? Hello?
Hello.
Am I audible?
Yes, sir.
Good morning, everyone. I welcome you all to this national webinar, the kind of national crisis, the language, the life of the patient now in the country. Thanks to the important persons joining in this webinar to accept the gathering along with the crisis. We have here today Dr. P.K. Officer, ADP, Deputy General of ICR at Sciences. We hear very much here and we see the topic. Great webinars. We can discuss the politics of how we ICR in this national. And we have Professor Virendra Singh, who is the coordinator, national coordinator of the NCI, Unnat Bharat Abhiyan, the flagship program of our Honorable Prime Minister, wherein the highlight education are taking the technologies to the villages in terms of rural health, rural development, and other livelihood to the area. I welcome you, sir, both of you to the board. And we have our scientists from other national institutes, like we have Manjula from Nivedi, Bangalore, and also Dr. Vinod area, who is a principal scientist from India, New Delhi. And we have our own scientists from Rio, that's the field, Anand Kumar, Bani Brahmala, and also scientists from various institutions of two medical university. Along with that, Dr. V. Padma President, also likely to join the so with this, now may I request Professor Vijay Sanjay, sir, please give you a valuable details of this crisis, wherein how the veterinary scientists are really active. And your main concern to the rural health and the rural livelihood. You wanted to save the farmers. That's why you asked us RPA, and that's the only veterinary university working with the UBA, and you asked us to deliver on national webinar involving the scientists to create awareness among the UBA participants. Thanks for giving the opportunity. Now, may I request Professor Virendra Singh to briefly tell about the physical activity, and then so your message. Then we will request the speaker. Professor Virendra.
Thank you, Professor Reddy. Now, this is very nice program being conducted by SVVU Tirupati. And in this program, which is very prominent for the country, for the animal husbandry programs, for the taking care of our animals in the country, and in collaboration with ICR, and I am thankful to Professor B.N. Tripathi, Dr. B.N. Tripathi, Deputy Director General Animal Sciences of ICR, as a chief guest in this program. Also the Vice Chancellor, आवाज आ रही है? V. Padmanabha Reddy, Vice Chancellor of SVU Tirupati. Eminent expert Dr. G.B. Manjunath Reddy, who is from Veterinary, Epidemiology and Disease Informatics Institute of ICR, and other important experts and colleagues. As you all may be aware that Unnat Bharat Abhiyan is a program of Government of India, Ministry of Education, in which all higher educational institutes of this country work along with some villages around them for their holistic development and livelihood generation. And
Hey,
this is a
Dr. Vijay,
हाँ
Dr.
जी
Vijay, I think Dr. Vijay, I think
या
two people are speaking.
हेलो
That I think two people are speaking simultaneously.
क्या करें क्या हुआ सर
Sir,
हेलो
I think there are two people speaking. Dr. Vijay, are you
अच्छा
also speaking?
ओके
We will ask them.
ओके ओके नाउ इट इज फाइन हेलो आर यू एबल टू लिसन
Yes, sir. Yes, sir. Okay.
Why is echoing? There's a lot of echo in the sound. But
yeah so this program is very prominent for the country and presently we are engaged with nearly 3,442 higher educational institutes, which includes IITs, NITs, central universities, ICERs, AIMS, IIMs, agriculture universities, agricultural colleges, and recently during the COVID pandemic, we organized web series for all the
Could you please,
fellow rural people of this country with our help of Regional Coordinating
Dr.
Institute of
Sarjan,
Unnat
क्या
Bharat
कर रहे हो
Abhiyan.
यार?
Yes, good, sir.
Every state has nearly one or two regional coordinating institute which are supporting Unnat Bharat Abhiyan and mentoring the participating institute of that state. I can give an example. In Uttar Pradesh, we are having three regional coordinating institutes. One is IIT BHU, another is IIT Kanpur, and third is the Dayalbagh Educational Institute University, deemed University Agra. Likewise, recently we had a program of Golden Diamond Jubilee celebration of IIT Delhi, in which Honorable President of India came on 3rd September in IIT Delhi. And during that period, she launched a Unnat Bharat Abhiyan success booklet.
Change the system.
आवाज आ रही?
And there is some echoes to man change the
We are just changing the sound system in within 2 minutes, and then we will start. हाँ नहीं नहीं मैं तो सर
Dr. Sarjan Rao, एक मिनट देखिए this is a very important webinar. I think two people are speaking together. So, नहीं, two people are speaking. Dr. Vijay is also speaking. Somebody else is also speaking simultaneously.
Okay.
So I think whosoever convening it or whosoever is comparing it.
एक मिनट में साउंड सिस्टम ठीक कर रहे हैं थोड़ा सा दिक्कत हो गई है
There is a lot of echo. हाँ पता इको आ रहा है बहुत ज्यादा उसको ठीक कर लीजिए कोई नहीं it happens sometimes doesn't matter
kindly bear with us yeah चला गया आ जाएगी इसमें थोड़ा सा ठीक है अब आवाज आ रही है बंद कर दो अब आवाज आ रही है? सब लोग
Okay, sir.
हेलो बोल रहा है
1 week in Lahore.
Dr. Vijay, I think you are logged in from two computers.
Two come. 1 week in.
बंद कर दो यार इसमें तो लॉकडाउन नहीं है हटा दिया मैंने हटा देता हूँ उसको बंद कर दो हेलो अब आ रही है आवाज
हाँ आ रही है
ठीक ठीक तो मैं I was just informing that recently President of India visited IIT Delhi in a program of our Diamond Jubilee celebration and released a booklet of success stories of Unnat Bharat Abhiyan. And the title of this is the book which was released by Honorable President of India and our Education Minister Dr. Dharmendra Pradhan ji. And the title of the booklet carries is Unnat Bharat Abhiyan, Transforming Higher Education and Villages for Atmanirbhar Bharat. And in that we have also received a message from the Prime Minister. I would like to read the message of our Prime Minister.
Dr. Vijay,
हाँ जी बताइए
आपकी आवाज बहुत इको कर रही है आपकी आवाज सेम आवाज भी इको कर रही है
स्टिल उसको बंद कर दो यार
Professor Reddy, is it with you also?
कोई दिक्कत हो रही है ना क्योंकि सेम आवाज
Okay.
हो रही है इसे आप
Is it okay from your side, Professor
Yeah.
Reddy?
Okay, pa.
और हम
So I request Professor Tripathi, please mute your. Maybe you have also started the YouTube.
Yeah. So our Prime Minister, the message which our Prime Minister has conveyed to me, I would like to read so that everybody working in Unnat Bharat Abhiyan can get a message that in next 25 years, what is the target set by our government of India for Unnat Bharat Abhiyan. So he mentioned that the completion in his message, Prime Minister mentioned that the completion of 8 years of Unnat Bharat Abhiyan is a momentous task, occasion, and greetings and best wishes to everyone associated with the program and to the Ministry of Education, IIT Delhi, and other institutions, as well as various stakeholders. Education and knowledge are pillars which support the nation's progress. We are striving tirelessly to make India a new knowledge hub and to empower higher education institutions to play a larger role in nation building. When it comes to development of our rural areas, we believe in Atma Gaon ki Suvidha Shahar ki. That was mentioned by our Honorable Prime Minister, which facilitate that empower aspirations and boost ease of living are reaching villages and serving the people. Unnat Bharat Abhiyan is an initiative that is playing the role of an enabler in transforming the rural landscape. The Abhiyan is pivotal in leveraging the intent and energy for our talented minds to devise solutions to accelerate sustainable and holistic growth of rural India. The Amrit Kal of the next 25 years is an opportunity to march ahead with the new resolve to realize the vision of a developed India in which education and rural development play a key role. Signed by Dr. Narendra Modi ji and addressed to National Coordinator Unnat Bharat Abhiyan, 1st September 2022. So I would like to convey that this is part of how we can empower our villages when they are facing some challenges. And recently, our animal husbandry system is facing a real challenge of lumpy disease. And I am grateful to SVU Tirupati University to organize this kind of awareness program for our rural people of this country, how to control lumpy disease in our animals. Thank you very much.
Thank you, Mr. Paredes. This is the use of how the media is working for the development, for example, the technology of ISP. So this RCI, SEG has got 113 prospect institutes, and every institute is adopted 5 villages, whereby they are almost reaching more than 500 villages. These technologies from CIE. Thanks for the initiative in the leader and the future areas of the known veterinary in the country, starting under the PPGA. Thank you for reading about the PPGA. So before my request the honorable speaker today, and I'd like to share some presentation on the basic information about the Lumpy Skin Disease, the scientific purpose that we made. So this is a very important national crisis we are facing. Can you see Rahul? Can you see the thing?
The people slides are visible.
Visible? Right?
Yeah. Thank you.
Yeah. So this is a topic that let me briefly view about the basic information about the Lumpy Skin Disease. So some of these parts include of RCA, also NCA. The Lumpy Skin Disease is the regular organization situation of after receiving using the in Afghanistan for the disease. So this is this is so this is the scientific how the antibodies are affected. These are some of the basic regions and the virus and the coming to the exam grade and also the evolution part. It's a Lumpy Skin Disease virus, the oxide something, temporary part. But we have other good parts also belong. Because somebody to avoid
First, negative control చేసుకుంటామా
how to use that. Yes.
ఏం వేస్తారు జనరల్ గా
Yes. The LFC virus, the pox virus of the pox virus, and other people go pox also belongs to the that's why initially to protect against LFC. So those parts is of the remedy to control the LFC or various information. In fact, this is erupted in Gambia and then afterwards the multiple outbreaks sometimes in only African continent. Later, 2015 reported in European Intradermal. The first time it was reported in 2019, when in some states of the Butler in the east of country. So later, 2016, after reported cases of European, many of the novel LSD vaccines are tried, but still the variant is mutating multiple so many variants and functions. This is taking the dynamics. So the major national scientists started LSD anybody but the function. So transformed in laboratory, then first the infection rate was only intel to some percent. It was not much of the infection, but people tried with that done. But the major issue came in July 2022, large outbreak came in Gujarat and Pakistan, followed by 11 other states, almost there, speaking like 1 lakh people. Then scientists reacted, and the the speakers, along with Granny Body, the publication article, he suggested that LMC virus may be able to evolve far into the bone. This is the problem. If it is by a vaccine. And Frank Van 2022, and they of science suggested that these outbreaks of yeah, are likely to be the result of some of the spillover from animal vaccine that is vaccine, electric vaccine. And so this is the seriousness of Lumpy Skin Disease. So almost all 15 states in a span of 16 months. Right. So this is the report from here now. So almost this is the valid information from the history of fisheries and animal participating. There are many lakhs of static successful growth and static that of and many countries are able to see. This created a serious of understanding. Coming to the basic information about the major symptoms, probably, or the constitution that is developed before. But initially, the farmers will mostly fever their then reduction in production. And you can find that a lymph node surface that shallow, which is very specific size to larger ones. And we should also notice in some of the findings that wherever the vaccine is tried, the lymph node size is the same. Then conjunctivitis, continuously, all these things. These nodules are all over the body within 40 years. So this is another observation, the end of part 9. The reason is that influential infection is also approved. The pregnant cow is aborted and not come to think of normal. The post is self-affected. The post-partum infection is really the worrying factor. Because many of the animals are so the same thing how does the disease transfer can be transferred commonly in a form where they are the smaller antibodies. Although we have a common source of feeding, so when the common contact to body changes with the share, so then by the transfer very easy and not animal, the other animals in the area is different. And sometimes from our interest like formally such as the so this is the infection carrying to the iron. Like I was mentioning about the Intra so this is also getting transmitted, but the infection can be the off in the wound distance. That's why when it's gone, it's also getting affected. Any out of it. That's why it's very essential to present the mother field in the pregnancy capacity. So other vector disease is whenever there is a flies and on the insect, there will be five of so one end of the infected animal is carried to the other healthy animal. That's why we need to prevent the vaccine them and also it protects the environment. Where obviously flies complication very quick. So these are the root of infections. The farmers have to be very cautious about the mode of it. So because the government is very conscious and the recently our honorable Prime Minister has taken this serious very seriously. The idea of all charity is protect. Then a lot of support from the investment collector from the JP board of council. I need to talk one thing about this economic impact. So this is a study from healthcare reported in science. So this is the perception from the farmers. Mostly the animal fertility, weight loss, milk productivity reduced, craft wherever the animal is scientifically craft, horsepower, terms of horsepower, reduced, animal productivity that you have. Mainly, there is high quality product, not just quality is for the cycling. Generally, the long stickiness in the movement position and abortions that able to thereby market get fairly. Treatment cost in terms of the animal cost ultimately the fertility and the problem. Because they continue to produce fertility of the very productive so coming to the other part, the comparison on the side, comparison between the exotic cattle or outbreak cow and outbreak material. Thinking of cows that are the inherent ability to resist them, and because of fixed things and they are able to fit to some extent, and they show this at outbreak cow. The outbreak cows are more affected than the exotic and individual that way. So of course, our DDT is playing more about the proactive. The vaccines developed by the groups are the country how you and the vaccination program is going on. And of course, area to the it belongs to Japanese virus. The both parts is for the same family, and the both parts is really because of this vaccine. So now we can just really talk more of the vaccination. Of course, many of these Ayurvedic and also other medicine, variants are reacting. They are studying very strong. And at topical level, it's about Ayurvedic, particularly food, vital, important. So this is basic information now, where it was undergoing DBT and what makes it. So the reporting this program, the reporting are totally valuable skills on the complete policies and also the IPR, which refers to the TDB. Sir, TDB, sir. First, sir. Yeah, please. What's here to look at that? Sir, sir, am I audible?
So you are audible.
Report, sir. Please go on the board, sir.
Professor B.N. Tripathi, please unmute your mic. Yeah. So you are unmuted, please proceed.
No, I have unmuted. हेलो नहीं हम नहीं हमको बोलना है वाइस चांसलर है ना इससे बाद मेरे पास वाइस चांसलर साहब नहीं है क्या ओके समझे क्या होता है चलो एनीवे मैं अपना कल चला जाऊंगा ओके I wanted to know whether vice chancellor is here or not.
Low voice, sir. With low volume.
Dr. Padmava Reddy is here or not?
Please go on, sir. But voice is low.
Hello? Am I audible?
Yes, sir.
Yes, sir.
No, it's audible, but the voice is.
No, I just wanted to know whether Honorable Vice Chancellor has joined this webinar or not.
No, sir, he has not joined, but please go on, sir.
Yeah, please. So that's why that's why I'm asking. Anyway, so very good morning and thank you very much. Dr. Vijay, so the problem is that my sound is echoing there. My sound is echoing, you know. If I'm speaking, same sound is coming to me back after a few seconds. And then how to speak?
Sir, the problem.
Please make sure that there is no other link open at your end.
No, this,
We have opened the YouTube also.
yeah,
YouTube
this
also.
problem is not from my side. This problem is not my side. This is problem is somewhere else.
Sir, please mute all of them except only.
No, I have not said anything.
Everyone is on mute. Only Professor B.N. Tripathi is able to speak. Rest all are on mute.
Problem कोई और हमारे
Professor Tripathi, please go ahead. There is no other mic is unmuted, so only you are there.
Anyway, let me start. So I think first of all that I really thank Dr. Sarjan Rao for inviting me for this national webinar on lumpy skin disease. I'm speaking. Yeah, I'm speaking.
Yes, sir. It's audible.
Somehow, yeah. Well, there's some problem here. Eh? So better you go ahead with that. Eh? Dr. Sarjan Rao.
Yes, sir.
I'm disconnected. There's some problem. Same sound is repeating again and again.
You can try with the phone link with that shared.
So I think you can just continue.
Okay.
You can just continue. Eh?
I'll continue then the answer.
I'm disconnecting. Please go ahead.
Okay. Now we take up the speaker and the on scientist, particularly Unnat Bharat Abhiyan. He's from Bangalore. I share ethics of veterinary analysis and basic information. UAV. So now I refer to each area of the chief molecular pathology and the epidemiology of provided area. So now I request Manjunath, please.
Just a minute, sir. I'm sharing, sir.
Dr. Manjunath, are you here?
Yes, sir. Just a minute, sir. I'm sharing my PPT, this one, sir.
Now to
Screen.
go for the video. Start your video also.
Yes, sir. Yes, sir. There is some problem. Just
You can mail us. We will share on behalf of you. Or either you can also share with Srajan sir.
just 1 second, sir. I'll. Yeah, it is open, sir. It is open. My screen is visible, sir.
Well, for all, please go ahead. Thank you.
Yeah, yeah. I'm audible, no, sir.
Yeah, you are audible.
Yeah. Thank you, sir, and good morning to all my respected teachers, and special good morning to Tripathi sir, who is my teacher, and VC sir, and the register and the dean, who have given me opportunity to talk about the epidemiology of this lumpy skin disease. So all of you might have observed this phenomena. This happens in case of especially emerging diseases, where we first notice that these kind of unknown symptoms are because in the field vets, anytime the disease starts as an unknown, then it becomes a unknown etiology of disease. One fine day, it emerges as a new disease. That's how the lumpy skin disease also happened when in 2019 in case of Odisha. So this was already almost covered, but I would like to emphasize that this is a more than 90%. It is an vector-borne disease, even though there are other routes of transmission. The economic losses also, sir has already highlighted, but my this one would be there are other impacts of this disease, like infertility in case of bulls, and even in case of some of the affected cows, and it has an impact on the traders, whether it is from a smallest trader like a Tata Ace fellow who is shifting the animals from one village to the even market or purchase. Once we ban the movement of the animals, no, there are n number of people in the chain of livestock management. They will also get affected. It's not only the farmer, and there are feed industry people. Again, these are the where it has an impact. And one more is I would like to add is because nowadays we are going towards the organic farming. If you look at the diseased animals manure, the quality of manure and the gases released by these diseased animals, they also contribute to the climate change. And the manure is going to be of less quality. Indirectly, it is going to affect whole of our agriculture farming also. That's my intention of showing this. And as you look at the history of the disease, it was almost a century-old disease. It was restricted to the African countries. And in 2000, afterwards, it started spreading to the other parts of the world. And 2019, we reported from India. And as on day, our all neighbor countries, whether it is Pakistan, Nepal, Bhutan, all are having this disease. And we had experienced 3 waves in India, one in 2019, 20, and 21, 22, and the present ongoing is 22, 23, where almost all the states of India reported the disease, except maybe Mizoram. Maybe they may be having, but still they have to report. And when it comes to the agent, because knowing the agent is also important, when you are giving for a news or press conference or even adopting the control measures, you please note that this is a DNA virus, and obviously the kind of mutations or changes what you expect in case of RNA viruses, you may not be able to expect in case of this DNA virus, and it is one of the largest DNA virus among the animal viruses. And it is very, very difficult to grow in case of cell culture. That is how even the isolation is not the mandatory for the confirming the disease. They go for PCR to confirm the disease. And it is an enveloped virus, hence it is susceptible to maximum, whether it is a chemical or disinfectants, which are alkaline or acidic in nature. They will definitely kill the virus because it is an enveloped virus. And it is also susceptible to high temperatures. Hence, we advise even the affected animal milk is safe for the consumption until unless it is not suffering from other mastitis conditions, because the boiling destroys the organism and it is a non-zoonotic in nature. The susceptibility definitely varies with the immune status of the animals, and there is only one serotype in this capripox or LSDV virus. We had also proved that the disease is or this agent, that is LSDV, is more closely related to the goat pox than the sheep pox. When you look at the evolutionary, this one, no, sheep pox was the first virus to be evolved, followed by the goat pox, followed by the lumpy skin disease. That is why the government of India has advised goat pox vaccine instead of sheep pox vaccine. Understanding transmission is very important. As of now, the disease is transmitted by maximum the biting flies. That is where your control measures are targeted for prevention or the disease for further spread. And even contaminated feed, water, or direct contact, they do play, but there is a minor role played by this. And there is a vertical transmission up to 42 days. The virus has been detected in the semen of the breeding bulls. Hence, if the bull is getting affected, it is advised that until it comes a PCR negative, the bull should not be used for the breeding purpose or for the semen collection. There is another mode of transmission is iatrogenic. If an infected animal, if you're using the syringes or needles, there is a possibility because it is a prick equivalent to a bite of an insect. And the long distance transmissions, whether between the districts or between the regions or between the states, this is usually attributed to the legal or illegal transportation of the cattle or vehicles. Whereas the local transmission, that is short distance transmissions, are within the village or neighboring villages, are mainly attributed to the vector population. Hence, we advise for the mass control or the prevention, we advise mass vaccination, whereas the local control, vectors plays a major role. Hence, vector control also should be taken care. There is no evidence of the virus being multiplied in case of vectors, unlike the blue tongue virus, where the midges are biological vectors, where the virus also multiplies. Whereas in case of LSD, in the vectors, the virus is not getting multiplied, and the virus gets killed within 7 to 8 days at the maximum if the vector does not find another susceptible host. If it sucked the blood from the infected animal, then transmission within 7 days, if it is able to find another uninfected animal, then only it can transmit. After that, the virus will die. It will not multiply in the vector. That is what I mean to say. It is a mechanical vector. And one more thing is that the transmissions are usually due to, as I said, congregations of the cattle. For example, in Karnataka or the other parts of India, where the bullocks are being used for the transportation of, for example, sugarcane. The sugarcane factory, if you look at, where more than surrounding villages, maybe 20 or the 50-kilometer radius, the all bullock carts will come there for deloading the sugarcane. During this process, even one bullock is getting affected. it can spread within a span of 1 or 2 days to the periphery of 25 kilometers. That is the one of the picture what I had shown here, one bullock with sugarcane carrying. Then susceptible host, obviously this it is highly host specific. As of now, cattle is getting more affected when compared to the buffaloes. Buffaloes, there is a milder form of disease, whereas in cattle you see very clinically pronounced disease, and morbidity, mortality, it is definitely varied. In the first wave, I'll show you some of the table in which we have shown. And the small ruminants, many times farmers do ask this question. I do have sheep, goat, and cattle together, whether sheep and goat are to be vaccinated or it should get affected. Obviously, because it is an host-specific virus, it affects only cattle and buffalo. So when it comes to the risk under the population, in India, we have a very good livestock resources. If you look at this resource map, there are 5-6 states where maximum cattle population is located. So these states are experiencing, or the disease is also very high. If you look at Rajasthan, for example, the numbers is also high, hence the disease is also high. In the same way, buffalo population is also susceptible. Maybe in next waves, you may see the disease in case of buffaloes, because it is the cattle is going to be herd immunity is going to be developed. And there are risk factors from the host side. Definitely cattle are more susceptible. Sex wise, there is no difference. Age wise, because young animals, they have only maternal immunity and they don't have well protected this acquired immune systems like lymph node or development of these lymphoid organs will take time. Hence, if a calf is getting infected, they are going to be severely affected. And the old animals also, because there is an immune system compromise, and there will be comorbid, like in many of the goshalas where you see there will be a comorbid, or they will be suffering from other disease. Because of this infection, they may die soon. They are supposed to die tomorrow, they may die today. Whereas come to the serotype, there is only one serotype. However, there is some variation in the this strains, but the serotype is same throughout the world as on day. And the virus, as I said, it is susceptible to many of the disinfectants and the chemicals. It can be killed easily, but in case of where temperature is less, the virus survives long time. And in the season, definitely wet and humid seasons, the disease abundance is more because the vector population is more. As I said, maximum the disease transmission is due to the vector population. And during the dry or cold seasons also, the disease is going to be less. However, during migratory season, like summer or during the fair or sandy seasons where the bullocks congregations will happen, there is high possibility of disease is being spread. Even the certain rituals like Bakri or these some of the festivals where they do sacrifice these cattle. So when it comes to the clinical signs, the first and foremost clinical sign you are going to expect is nasal discharge and lacrimation, and sometimes the edema of the dependent part. The edema is usually under the body parts, that is ventral parts of the body. Whether it is sometimes bilateral, sometimes unilateral, even the lymph node also, sometimes it is prescapular, sometimes it is prefemoral, we have seen. Even the lymph node only one side gets enlarged. That is how the disease presents. It is not unique in case of all the animals. Even within the herd, if you observe, each animal with the same farmer also, the disease presentation changes. If it is an lactating cow, there will be up to 70% reduction in the milk yield. And in that lactation, it is not going to come to the normal peak. For example, if we always calculate for the duration of the disease only. So you see, for example, 3 liters it is decreased during the 7 days. That is not the way to calculate the loss actually. The whole the lactation period, because it is not coming to the 10 liters back. That 3 liters is gone throughout the lactation. That is how we have to calculate. Then there is an temporary blindness and sometimes the ulcers. Nowadays, we have seen in the third wave, lesions are generalized in nature, and even on the gums and tongue and nostrils, we are finding the disease, and it is getting complicated with the bacterial infections. If you look at morbidity and mortality patterns between the first wave, second wave, and third wave, the morbidity is almost same, whereas mortality it is getting increased from first wave to the second wave. And the form of the disease, in the first wave we could see maximum it is dermatitis form, and it was not much respiratory system was involved. In the second wave there were deaths due to respiratory form. In the third wave we are seeing the whole of the organs are getting infected, whether you take liver also, you take even I'll show you some of the photos where gallbladder is also showing the lesions on the tongue. Sometimes you may not have seen such kind of pictures. So this is the early clinical sign. One of that, there is a possibility of easily getting confused with either thylaria if the skin nodules are not shown. Sometimes lymph node enlargement is getting confused with the thylaria. And obviously we are also finding some of the 25 to 30% of LSD cases are also positive for the thylaria. And the dewlap enlargement or the edema is getting confused with the fever, HS. So you have to differentially diagnose this disease. The skin nodules will be definitely pronounced in case of less hairy breeds or the white-skinned animals. You can easily see. And the nasal discharge and this one, no, without any skin nodules, you may get confused with the nodules in the mouth with the ulcers, maybe FMD kind of symptoms. This is a mild stage where the numbers are less when compared to the other form where the disease is going to be spread. This picture speaks about the probability of survival of animal. What we have observed is, the field veterinarians from their feedback, if the nodules are open, the probability of animal being course to the morbidity or mortality is less. If the nodules are big in size and the probability of disease succumbing to the death is more. Maybe due to this one, no, release of these inflammatory mediators, all those, the animal is relaxed. In the same way, if you look at the size of the nodules and the numbers, the number of nodules are more in case of domestic animals or indigenous breeds when compared to the crossbreeds. Crossbreeds, they do develop the disease. Both are equally susceptible, what we feel, indigenous as well as crossbreeds. But in crossbreeds, because we do take care of them with a lot of, whether it is giving bath or giving good nutrition, providing health care, all these things, because they are giving you milk, there is a high probability of less deaths in case of crossbreeds compared to the indigenous breeds. There is a secondary bacterial infection. We advise not to give the bath in case of later stages if the nodules are very big, because always there is a secondary bacterial infection followed by some fungal or infections. In case to buffaloes, what we have seen is a very milder form, and it goes unnoticed, unlike where the cattle, where you find the size of the nodules and all those things are very big. So it is unnoticed, and maybe the species are more susceptible. Such kind of studies are still lacking. And we have seen mastitis complications as well as blindness. And one more, the most complications with the LSD is if it is open wounds, the infection with the maggot, this one, you have to apply a lot of fly repellents and whatever ointments or even homemade therapies. For a veterinarian at the field level, what is the sample to be sent at different stages of the disease? If it is in early stage, because if you want to control further spread of the disease, diagnosis of disease plays very, very important role, and detecting at the earliest possible stage is possible only through swabs and blood, where even before the development of skin nodules, we can detect the virus in case of nasal swabs or lacrimal swabs. As the disease progresses to mid and late form, that the clinical signs of dermatitis will be evident, and you may take skin biopsies and scab. Please don't wait for the scab to develop. When the scab is developed, means already it has gone up to 3 to 4 weeks or up to 5 weeks of the disease. It might have spread to the other susceptible population in that village or to the neighboring villages. I have given also what are the preservatives, what is the temperature. Swabs can be collected in any kind of viral transport media. Blood, whole blood is needed. Serum is not the sample, clinical sample to be sent, because it is mostly a CMI mediated disease. Even if you send serum also, it less serves the purpose. And what we are doing at the labs or any kind of presently, DADHD recognized veterinary colleges also, some of them, the PCR is the main thing they advise, whether it is conventional or real time. And we do meantime virus isolation also. We are doing the immunohistochemistry, agglutination, and even ELISA and VNT and dot bloods are also being carried out. Some of the post-mortem lesions, what you are going to observe in case of animals died due to LSD or suspected of LSD is through and through skin nodules and pock lesions in the respiratory system. There will be very pronounced kind of this one, and gunshots, that's sheep and goat pox, what you see, the same kind. And what we have seen is also this trachea upper portion, larynx, it will be a gunshot wound kind of lesions. And when you cut also on the surface, you'll find such kind of lesions. And on the gums, if you look at it, is sometimes you get confused with FMD. And the tongue also, you see the kind of lesions, circular and pock lesions. When you do histopathology of these biopsy samples, hydropic degeneration is the more evident form of the degenerations, where due to this, the death of the cells will happen, followed by infiltration with the because it is a viral disease, the lymphocytes plays a major role. But if it is a scab one, definitely neutrophils and secondary bacterial infections do play. The one more, what we have observed, more than 100 animals, what we did, we are not unable to detect inclusion bodies. That may be the one of the typical finding in Indian LSDV strains, whereas in other part of the world, they did detect the inclusion bodies. As of now, we are unable to detect. That may be one of the peculiarity of this virus, which is Indian strain of LSDV, that's why it is more pathogenic maybe. When it comes to therapeutic management, definitely there is a course of antibiotic needed depending on the form of the disease. See, we have 3 forms of the disease as of now. It is a dermatitis form, respiratory form, or generalized form. So based on your experience, what is the best available antibiotic available in your region, you have to take care of that. Whether your approach, whether a veterinarian can travel to, because each veterinarian may be burdened with a lot of animals. So sometimes you may not be able to travel to the remote areas. At that time, you may go for long acting. Where you are easily accessible, you may give the antibiotics accordingly. Because the skin is one of the biggest organ in our body, so there is nodules will cause a lot of pain. Even we have seen some of the animals, they are standing for almost 1 or 2 days without lying down because the edema caused and the nodule pain. So you have to give anti-pain or painkillers and antihistamines. And because the mouth nowadays, as I said, the lesions or ulcers on the tongue and mouth, the animal is unable to chew the hard, coarse materials. Hence, your request or you can advise the people to give easily digestible or liquid form of diets are also advised. And immune boosters and any minerals, all these will definitely help them to combat the disease at the early. Sometimes even homemade remedies like herbal medicines, all these can also be advised to combat the disease. And there are diseases which needs to be differentiated. It is easy. And these diseases, they don't cause such kind of generalized skin nodules, whether it comes to pseudococcopox, insect bites in urticaria, photosensitization. They respond to antihistamine treatment in 24 to 48 hours. The nodules can be not be found everywhere. In the same way, when it comes to onchocercosis or this parasitic, they cause in the ventral portion of the umbilical region where the nodules are there, not all over the body. bovine stomatitis, it is restricted to the mouth part. It is easy to differentiate with the others, but you have to consider. When it comes to the prevention and control, the only one method to control the disease is we have to go for vaccination, followed by the restrictions, whether it is animal restriction from one region to other region, or within the local in terms of isolation or quarantine, and avoiding communal grazings. All these comes under the implementing the restriction on the movement of the animals. Then comes the third is vector control, which is again in the locally the spread is, as I said, it is due to the vector population, and vectors are also complicates the wound by maggot, all these things. Hence, you are advised to go for repellents or even sometimes insecticides. They do spray on these farms, and farm level biosecurities, like as I said, isolation of the animal, or cleaning, or keeping the health of the animal, giving good food. All these are concerned with the farm level biosecurity measures, and proper carcass disposal is also one of them. But out of these, for farmers, if you make them aware, whether it is a veterinarian, any stakeholder, you please make them aware what is the disease exactly. If you make them aware of any disease, they say 50% of disease is controlled when you exactly know what is the disease. With that take home message from my this one is would be, if there are no disease, you please keep a regular monitoring, or keep a vigil on the where unaffected areas are there, and vaccinate the susceptible population. You know better, for each state there is variations, and the area will also vary. Some areas where high dairy production animals are there, you can go for preventive vaccinations, all these things. Then creating awareness among all the stakeholders, whether it is line departments or police department, because when you implement restrictions, you have to take into all these people, from IAS to the police department, revenue officials, all, even human hospital, all these people will come into help, because many people will ask you whether it is zoonotic or not. So then preparing SOPs at the regional level, depending on your capacity, you have to advise them. You cannot follow a common guidelines from the national level to the regional level. You have to modify according to the requirement of your this one. And preparing the RRTs, that is rapid response team, which is definitely, it should include what I feel, the master's degree holders or youngsters who can move fast and address the solutions. And when you are expecting the disease, before that, one of the preparation is vaccine and medicine stock, keeping them, and networking with the other people. So you can expect, even if it is subsided this year, you may expect next year it may going to be an outburst. With that, I thank you for giving opportunity. Again, I thank all the organizers as well as my teacher Tripathi sir for giving me an opportunity to share my knowledge.
Thank you,
Over to you,
Dr.
sir.
Thank you very much. Very comprehensive and very points to the interesting. It will not required for the scientist farmer to research the I request you to be here for taking the questions.
Sure, sir. Sure.
Dignity sir is like to speak after without the area. So, will you be with us? Are you going to be with us? This is the end.
Yes, sir. Yes, sir.
Yeah, thank you. Now, may I request the speaker, the important speaker, actually, the PSI of Institute of Veterinary Integrated Biology University.
Manju, Dr. Manjunath, just one intervention.
Sir.
See, you said that in human body may not be present, may not be the characteristic of this virus present in India. But perhaps I have seen some slides from Gujarat. They have got beautiful infusion bodies. So that could not be the differentiating feature, right?
Right.
Thank you. We have to please go ahead, Dr. Vinod.
Okay, thank you. So, may I request Luke David. is a principal scientist from CSIR in integrative biology. He's a clinician and a computational biologist. His laboratory is interested in finding that the function organization regulation of particular genome and how genome variations could potentially impact us. He's also involved in creating novel methods and resources for analysis and annotation of the genome and understanding the functional effects of genome variation. So he has done a lot of work on this genome work of Lumpy Skin Disease. Now may I request Vinod sir that unmute your. It's not audible. Please unmute.
Am I audible now?
Perfect.
Yes, you are audible.
Yeah.
Please go ahead.
Thank you very much. So I am probably the odd man out because I've never worked in animal diseases before, neither does our institute work in the area of animal diseases. I come from the CSI Institute of Genomics and Integrative Biology, and most of what we do is in the interface of clinical medicine and translation. We work a lot in the area of genomics, where we try to use our skills to address very pertinent problems in human health. But this is probably the first time we have ever ventured into looking at animal diseases. Now, nevertheless, the principle of human and animal diseases, or in terms of microbiology, is very pertinent and very common, because pretty much everything that we work in microbiology is indebted to this one man, Anthony von Leeuwenhoek, who was a scientist by hobby and by profession a merchant and a municipal official. And his claim to fame was this microscope, handheld microscope rather, through which he could observe the microbe world around him. Now, almost 400 years later, this microscope, of course, in multiple different varieties and forms, today we use an electron microscope, has been the mainstay of looking at microbes. But nevertheless, there have also been a sort of silent revolution happening beside us, and that is in the area of genomics, and that's where I come from. And the consistent increase in throughputs and the consistent reduction in the cost have enabled a large number of genomes to be sequenced across the world. This includes a wide variety of microbial genomes, a wide variety of pathogen genomes, and of course, a wide variety of host genomes, including humans and animals. Now, what the technology has changed in the last 5 to 10 years has been that this has also been implemented directly in microbiology. Now, for example, this is one slide which sort of pictorially represents the time taken to investigate an outbreak. For example, the previous Ebola outbreak, which happened in Africa, took almost a year and a half for an accurate characterization of the virus. But that is not true anymore, because you can take the sample and directly proceed to genome sequencing to identify or molecularly characterize the particular pathogen, whether it is a bacteria, whether it's a virus, whether it's a parasite. You could today do direct analysis and molecular characterization with genomic sequence at a fraction of the time that conventionally it took to characterize a pathogen. Now, of course, that would also mean that you could understand the pathogen at much greater depth. And today, the question in focus is essentially a virus which belongs to the Capripox virus. The previous speaker has sort of alluded to it and explained it much greater detail. And just to give you a brief glimpse is that there are two pox viruses today that we have been talking about. One is, of course, the monkeypox, which is affecting humans, which is belonging to the orthopox viruses. And today we are talking about Capripox viruses, and here very specifically the Lumpy Skin Disease virus. Now, in genomic similarities, they are quite similar to each other with pretty much the same genome size and pretty much a conserved set of genes between them. But of course, there are differences and very specific in terms of the host. That necessarily means that they don't typically jump across hosts to become a zoonotic disease that easily. Now, what we know about the Lumpy Skin Disease virus is from the genome that was sequenced in 2001. And this was from the Neethling strain isolates from Africa. And this was originally isolated in 1958, re-isolated later in 1987, and was retrieved from an experimentally infected animal. And now what we know from this particular virus is that the virus has a very conserved central core and a repeat region across the two terminals, what we call as the terminal repeat region. And almost all of these genes are pretty much conserved across all pox viruses, whether it is monkeypox, sheeppox, so on and so forth. And what we know today is that it has a 151 odd base pair genome with an inverted terminal repeat, as I stated before. It encodes for 156 odd protein coding genes, and there is a very high similarity between pox viruses. And as we know today from the pox virus evolution, pox viruses being DNA viruses have a very small rate of accumulating genetic mutations, and largely the evolution is directed by deletions or genomic recombinations. And these two are very important even in the present context. Now, of course, the previous speaker alluded to this, but just to reiterate the fact that until 1980, this was largely an African disease, but it's only recently that it is spread across different parts of the world. And to be very specific in this particular outbreak, in just 3 years, it has affected close to around 2 million cattle, causing around 100,000 cattle deaths officially. And so we worked with the State Disease Diagnostic Center from the Department of Animal Husbandry, Government of Rajasthan. And we used a standard sequencing approach, which is called the shotgun sequencing approach, which doesn't require any a priori isolation or purification of the particular virus. You can take the sample and you can grind up the sample and sequence it directly. Every single piece of DNA, including of the pathogen and the host, is sequenced. And using extensive bioinformatics analysis, you could sort of retrieve the genomic sequence back from the mixture of samples. And of course, we have done this quite extensively previously in the COVID pandemic, and also to investigate multiple other outbreaks. And we also now extend this to looking at the lumpy skin disease virus. And now what we got from this particular virus was quite surprising, because as I said before, the lumpy skin disease virus, being a pox virus or a DNA virus, has a much lower rate of accumulating genetic mutations. But in this particular case, we found a whoopingly large 170 odd genetic mutations compared to the Neethling strain or the reference genome of the virus. We also compared the genome sequence, which was sequenced from the 2019 outbreak in India from Ranchi. It's called the Ranchi isolate and is deposited in public domain. And nevertheless, this number of genetic mutations were whoopingly high, even compared to the previous genetic isolate of the virus. So in brief, we had 8 DNA samples from the skin scabs as well as nasal swabs from 5 animals. At least one animal had 2 samples, one from the nasal swab and from the skin scab. What we found was that this particular genomes had 46 unique mutations, which are not present in any one of the lumpy strain genomes. There are around 56 odd genomes available in public domain. These variants are never shared with any one of the previously sequenced isolates, and out of which 39 were present in all the 6 genomes, out of which of them were non-synonymous and others were insertion deletions. Now, this is very significant because, as I said before, the evolution of lumpy skin disease is largely determined by genomic deletions as well as recombination events. We have not yet observed a recombination event, but as the previous speaker sort of alluded to, that is something that we need to be looking out for, because in every large outbreak previously, either in Russia or in China or later in Vietnam, there have been recombinant viruses which has emerged, where the recombination happens between the pathogenic strain of lumpy skin disease and the homologous vaccine isolates. Now, we could put that back into a genomic cluster, and what we realize is the genomic cluster, it stands out quite similar to the previous outbreaks in the Central Asia, including in Russia and Kyrgyzstan. And in terms of the closest similarity with other genomes out there, they stands out as a very distinct lineage or a clade of virus. So what we know today is that this particular genetic lineage of the virus, do not have a close ancestor. They are similar to the Central Asian outbreaks which has happened before, but nevertheless do not have a close relative, even quite distant from the older isolate from 2019, which was sequenced from Ranchi. The other surprising thing that we sort of observed was the intra-host evolution. As I said before, pox viruses do not accumulate genetic mutations very rapidly. In this particular case, we did have 2 samples from the same animal from 2 different sites, from the skin scab as well as the nasal swab. We do not expect to have a genetic mutation within the host because of the low rate of mutations. But in this particular case, we did find a low rate of mutation, at least 1 mutation, which accumulated between the 2 samples. Now, this is very surprising. Of course, this is an n equal to 1. We need to probably do a much more number of samples to sort of ascertain why this is happening, and also understand what is the driving force of this large number of mutations that we presently see in the Lumpy Skin Disease virus. So in summary, from the genome sequence, we identify a distinct cluster which doesn't have much of a similarity with the previous isolates from India and across the world. We have 47 distinct mutations, and we also suggest potential intra-host evolution, which suggests a much larger rate of mutation than previously characterized. Now, that also means that we need to do much more than before. And what we need to do is probably do a much stringent and spread out genomic surveillance, because that will allow us to understand much more about the introductions, the patterns of spread, the evolutionary history, and more importantly, to track emerging variations with recombinations and deletions. And this is extremely important in the genomic perspective, as well as also in the preparedness perspective. Having worked extensively in the COVID pandemic with viral genomes, genomic evolution and tracking new variants is extremely important, especially in an emerging pandemic. And this is even more importantly because there are a high level of mutations that have been presently observed, and there's a distinct clustering of the genomes with significant diversity from the previous genomes, and a much higher morbidity and mortality observed compared to the previous outbreaks in India. Now, of course, the technology for this is presently available, and this is one such technology we piloted in 2020. We were one of the first clinical sites to actually test this technology, which is today a global de facto technology put together by Illumina. And the advantage of this technology is that you can do scalable sequencer right from benchtop sequencers, the smallest of the smallest sequencers, to the largest of the largest sequencers without a change in the chemistry. You can scale anything from 48 samples to around 3,000 samples in a very modular and very cost-effective format. Just to put in a price, it would approximately cost around 5,000 rupees to sequence a COVID isolate of the virus. And this has been extensively used in India as part of the INSACOG, as well as many other countries which run genomic surveillance programs. And today we have a compatible technology like this for the Lumpy Skin Disease virus, which we would be more than happy to work with any interested partners to actually roll out a genomic surveillance program. And really more important is to understand why we need to really run this genomic surveillance program. Is not just to say that the wave would be over, but to also look at coming up the waves that are going to come up in terms of early identification, in terms of estimating the origin and spread of the outbreak, in terms of developing healthcare interventions, in terms of tracking the prevalence, monitoring the evolution, and more importantly, to train the next generation of individuals in genomics and data surveillance. And of course, to automate all of this, during the COVID pandemic, we did develop a computational pipeline, which is extensively used in the Indian surveillance program, as well as the Southeast Asian surveillance programs, which pretty much automates every aspect of genomic surveillance. This will also allow rapid scale up of genomic surveillance that, as you would see, had happened during the COVID pandemic, with today over around 58 labs across the country rapidly latching onto genomic surveillance. And this is today possible with large organizations. For example, the ICAR, the DG of Animal Sciences, is in here. And this could pretty much be considered for rapidly expanding the genomic surveillance programs. And what we're really need is, of course, an intelligent system. We still had lost a significant amount of time in trying to control the outbreak. So it's really more important to use digital tools to track and trace the outbreak. And that's a lot of learnings from the COVID pandemic. And of course, how can we use molecular approaches to rapidly analyze and rapidly characterize an emerging pathogen? I'll stop there and take questions. But before that, let me acknowledge a lot of people who made this possible. Dr. Lenin Bhatt and Dr. Ravi from the State Disease Diagnostic Center, and my colleagues at IGIB, Dr. Sridhar, Dr. Rahul, Dr. Bani, and many others. And the funding for this came from CSI. Thank you very much.
Thank you, Dr. Ravinod, within the wonderful information of research paper. Thanks once again for opening the universities and opening for the networking with the relationship that I are now to take the university. So we have already talked to you about our own time system. So I don't think develop network abilities that, the sequencing abilities that really do the way for that important. So I request you to be available for the future take from the next after the evening. Please. Dr. Vinod, will you be here to compile?
Yes.
Thank you. Now we are waiting for the speaker message today. So now I request what is the valuable material that but thank you for spending a lot of time with us and waiting for long. Please.
Thank you, Dr. Rao. And thanks for inviting me. In fact, this is a very, very important webinar because this disease for the last 3 years, especially the last 6 months, has really devastated the country and the cattle population especially. And throughout the country, many national webinars are being conducted almost weekly, I'm seeing, to create an awareness among the people that how this virus could be dangerous. Though Dr. Sarjan Rao and Dr. Manju and Dr. Vinod, all three have given pretty much account of everything about the disease characteristics, how the disease and the historical perspective from where south of African countries, how it has come to Middle East, and then finally it has come to Indian subcontinent. I'm audible to all of you?
Yes, sir. Perfect.
Yes, sir. Yes, sir.
So finally it has come to India, and during this year only, it has been so devastating that it has caught attention of people around, and people are worried that we have been thinking that the disease causes only morbidity, maybe around 40 to 50% morbidity, and 2 to 3% mortality. But this time it has caused really, maybe around 10 to 15% mortality, and more than 1 lakh cattle have already died, and more than 20 lakhs almost cattle have been affected with this disease. Certainly that if you look at the disease, the history from 1930 onwards, it has been confined mainly up to 1985 to African countries, and from there it has come out. So Middle East and then South, this Russian Federation, Balkans, Middle East, all these countries, and finally when it reaches to India, in Bangladesh and other neighboring country, it is also creating similar problem. Well, this is captive pox, that has been told that it is quite close to sheep and goat pox, and sheep and goat diseases are already there in the country. And I would like to speak something about on the vaccine aspect, because both all three of them, they have not spoken about vaccine. Normally, the countries where we do not have any homologous vaccine, and in such cases, this is either sheep pox or goat pox vaccine are generally, permitted, so that at least they provide some cross protection, and those cross protection are pretty good also. And if it is done religiously, sincerely, with very good biosecurity, it almost gives good protection. But in the Indian situation, it is very difficult. And what reports we are getting from different farms and gosalas where the goat pox vaccine was there, after 7 to 8 months, disease has occurred. Okay. So, and some mortality is also there. So certainly it is not going to provide a foolproof or 100% protection. Now, when talking of the vaccine, what is available in other countries, this needling strain is one that has been available, a vaccine from this, and several companies they are selling out of this vaccine candidate, and also Kenyan sheep and goat strain. They are also there, and our Indian strain is in between needling and also this Kenyan sheep and goat KSGP strain. Some variation, as Dr. Vinod told that there is some mutations available, and so really we need to keep a very good watch on this by genomic surveillance, which is required. And we never expected, in fact, for the Capri pox or the pox viruses, they are so stable, so heavy, more than 150 kV. So we don't normally expect it. We expect it more in RNA viruses, but here we are getting some variation. Though his study is based on only 5-6 samples, but even then it is eye opener that we should go really for the surveillance, and this is very important. Now, when I was talking about this vaccine, then luckily when we got isolated from Ranchi during the early, we thought that let us go some value addition to this strain, and we could compare the attenuation process that has been used in different strains, for example, needling or KSGP strain. They're quite different. And there has been a problem with needling strain also. We get around 10 to 15% cases of swelling, sometimes abortion, some reaction also. And so that's why people always suspect this vaccine wherever it has been used. And when it was used in Russian Federation, in Kazakhstan, there's no. And one thing I would like to inform all of you that there has been no natural recombination, right? We just normally, spreading sometimes, rumor among the people that there is no natural recombination of the virus. So the new virus has not come up. The Russian Federation experiences where vaccine was banned, it was the vaccine seed culture was spurious, where when they tested 2 batches of vaccine used in Kazakhstan and some other country, and then they caused disease. And they, when checked these randomly 2 batches, they found that they have got several, they have got sheeppox also, goat pox also, LSD virus also. So perhaps somehow there have been some mixture during the seed culture preparation this has happened. So one thing is that very clear that till date we do not have natural recombination. And there's also a feeling that if you use LSD vaccine, then it may create homologous recombination. So perhaps that probability is quite remote, as the virologists and people have been seeing. And till date, there is not a single vaccine, attenuated virus vaccine, where it has there's been recombination, it has new strain has come up. Whether it takes sheeppox, goat pox, rinderpest, even PPR, all these viruses, we have never come across, and people know it. Then coming to the vaccine production that ICR has produced, this is very important. Luckily, unfortunately, this vaccine has been doing so good that is not causing any adverse reaction. There is absolutely no abortion, no swelling, not much of milk reduction, not much of fever, no local swelling even. And it has been tested in almost 25,000 animals in field data, leave aside experimental data that we have done in IVRI Venkateswara. So this vaccine, we have been found it is working very well. And when we are working on its sequencing aspect also, we have found that somehow more than 800 nucleotides have been deleted, and those deletion have not been able to find in needling strain or any other strain. Of course, there are some mutations in 6 to 7 genes, that has occurred in all these vaccine strains. But our strain, our virus vaccine, they've got more than 800 nucleotide deletion. So that gives perhaps, luckily we have got it, a very good vaccine candidate, and that's why it is working very well. And we are also, ICR is working to look at that, what is the protection ability of goatpox vaccine. And then how we can, when this vaccine has already been commercialized, and one company already taken, they've got a license, and they're going to produce it very soon. And then other two companies also they have also trying to take it from ICR into vaccine. So that vaccine availability, normally we require 300, we say that we have got 300 cattle and buffalo in the country. Around 192 million cattle and 109 million buffalo. Buffalo, normally we can avoid it currently because it is only mild disease. But certainly cattle, 192 million cattle are the state. They're susceptible. Crossbred or indigenous breed. As Dr. Manju has told very clearly that there's also a rumor that our indigenous cattle is more susceptible than the crossbred cattle. But perhaps it is not. There may be possibility of some severity, maybe size of nodule, size of convalescence process, but certainly there is not much difference in that. Especially all naive animal, young animal, and old animal, they are more susceptible to disease. So now coming to other aspect, that biosecurity is very important. Along with the vaccination, somehow today after 2 to 3 months, when vaccine will be rolled out in the market, then we'll have to switch over to lumpy skin disease vaccine, and perhaps we can able to control the disease within the next 5 years if we do it, on a very integrated manner and intensive vaccination of all cattle, and perhaps we can able to achieve it also. Biosecurity is very important, and as all of you have really very detailed that how we can prevent and control the disease, and also the treatment process, palliative treatment, symptomatic treatment, that is also very important, and we can really work on the save our animals. Now, when you do the postmortem of such like animals, you find, as Dr. Manju has told, that all nodules, these nodules and ulcers, they're all found throughout all internal organs. And some of the photos, picture I have got it from Rajasthan and Gujarat, they're really horrible. And then how these animals could have been suffering from external lesions also, internal lesions also, and therefore there is no chance of saving such like animals. So the take-home message from my side is that we have to really work together along with scientists, veterinarians in the country, and other aspect that Dr. Vinod has very clearly put out, that he has got 47 mutations. So that is also, we have to keep a watch on that. We can go for some genomic surveillance throughout the country and see that how this virus is evolving. And if it is not conventional type, that we are thinking that capillary pox or all pox viruses are very stable, but we are getting such like mutations very frequently. Today it may not be important, because when we checked our virus, our vaccine, and today's vaccine sera, and this vaccine is absolutely protective. So it appears that it is not a major entering variation. Serotype is only one, some minor entering variation, but it is not converting into something a new serotype, and therefore the currently available vaccine, which is working very well, whether in 2019 strain or 2022 strain. So then I would say and advise that all of you that we have to recommend this vaccine. We have to go with that, because there is always two school of thought, since goatpox vaccine is already going on in the country. Many people are not adopting it. They're asking for the LSD, and there are also other part of other school of thought is that LSDV might create problems for new recombination miracle. So such type of feelings and thoughts would not prevail throughout the country, otherwise it will be a problem. So, the take-home message is that as long as we do not have any LSD vaccine, we should go for this goat vaccine or sheeppox vaccine. But when we have LSD virus homologous vaccine, we must go for it immediately as early as possible. That is the message from my side. So thank you, Dr. Sajan Rao, and all distinguished speakers for also increasing my knowledge. And certainly, Dr. Vinod and others' advice, Dr. Manju, the advice will always be there for all of us, and people will really, work on it and see that this disease should not be taken very lightly. So very lightly. It has been in African countries for almost 60 years, and there has not been much research also on this virus. That's why not a very good vaccine was there. And fortunately, luckily, incidentally, we have got a very good virus candidate, virus vaccine of LNT, and that must be used in the country as early as possible. So with this, thank you very much.
Thank you very much, sir, for your everyone very informative information on the vaccines and the right. Definitely, sir, you have given a very clear pinpoint advice on usage of the vaccine that is for the right here. Still, that the discussion is going to be they all thank you. We always contradicted one to other. But you also clearly made a point that the vaccine available that cannot be used now right here. Really good one, potent one, which we can really use. That's why we also limited for the vaccine, and the concerned person said to us that the vaccine is very much available. Only with the conversation they cannot take as much as possible. The vaccine is available. Thanks for the for taking the initiative production of the vaccine. They are very specific elected. So definitely we play for the immunity forms and also the related department to make the organs right. And also this definitely this webinar will open a good networking program for the secure that we can also take agriculture in the case. Mainly on the genome, the genome discussion, there very good open information which is the need of the hour on the IDC of the details. Where they say more than 40% especially which is putting on those lines. But you have given a valuable time. I know how quickly you work. When I visited you, define lines to compare you, but in spite of that, you are making our time and percentage that policies and also the government of India conference on the definition of village. Thank you very much from UBS and also from the city side. So for your participation. So we have a question that we can phones are available. Thank you very much. Now the SUBU has got some potential. We have got good cycles. Even 2019 itself, we have diagnosed some of the types of LSD. We have 3 scientists and some more scientists also in the interactive. First, we have a state level diagnostics laboratory that Tirupati headquarters, there's in Dr. Anita and the earlier scientist Dr. Vakit, and after we have Dr. Anand Kumar from the PGR College. We also published valuable information from Dr. Vakit. So now may I request that Anupramila, what exactly your experience of 2019, what are the period about the genotype, and also what are the diagnostic measures they have taken in 2019, but we are not taken forward to the vaccine production, but we can give a promise that we can go for the vaccine production. Now I request Dr. Rani Pramila, please speak that quickly, then we will move to the other. Dr. Rani Pramila.
Yeah. So thank you, sir. So good morning to the dignitaries, VDG Animal Science, Dr. Abhiyan Tripathi, sir.
You may open your video.
Yes. And the dignitaries, senior professors, heads of research station, all the professional colleagues. So good afternoon to all of you. So I thank the organizers giving this opportunity to present whatever the research were carried out at a state level diagnostic laboratory as a part of research station of Venkateswara Veterinary University, Tirupati. So since 2019. So thank you, sir. Yeah, of course, this is our university, Venkateswara Veterinary University at the foothills of Lord Venkateswara. And this is our state diagnostic laboratory. So apart from the all facilities, we are having the unique facility of BSL-3 facility in our lab. Because of this BSL-3 facility, we have done all pandemics of COVID duties, and our lab is one of the center for the doing COVID diagnostic center during pandemics for 1.5 year. And as far as lumpy skin disease is concerned, actually in the Andhra Pradesh, so earlier speakers told the significance and economic importance of lumpy skin disease. So initially it occurred as a sporadic in 2019. So what Dr. Manjuna read, endemic, and worldwide it is become endemic from 2019 to 2020-23. So initially it will occur as a cutaneous form, and later it is becoming including all the systemics leading to the generalized systemic form leading to the high mortality. So as far as Andhra Pradesh is concerned, so initially have seen only in the certain districts what I will show the data, and now it is spreading almost most of the districts are showing the symptoms of the disease, and the disease has been diagnosed. And as far as in Andhra Pradesh, this is mostly seeing in the cattle. So we are not, I think, as far as our research, we are not collected any samples, or we not received any samples from buffaloes. So what are the samples we are collected is only from the cattle and shown the clinical signs. I think why we are so much of importance in the lumpy skin disease, because of economic loss, both in as well as production loss, as well as skin losses, and because of these things we have the importance of the disease. Now we are very curious about this lumpy skin disease. So earlier we are not knowing, when we are studying in our college days, lumpy skin disease, that it is only one of the genus of the capripox virus. And as a teacher, I have shown the students only the photo picture. So whatever our teacher brought from the states, I have shown that lumpy skin disease doing generalized skin lesion throughout the body. That only the picture I have shown earlier days when I was teaching to my students. Now the same disease, I said that during that time this is only in the states, not in our India. So I said at that time, but now so it become more endemic in our India and including our state. So that is the scenario of the disease changing from day to day. Yeah, as far as samples are concerned, detailed samples, we have collected only blood samples as well as skin scabs from the respective animals showing the clinical signs from the cattle. And these samples normally we are collected from different districts of Andhra Pradesh at that time where outbreaks are occurred. And initially all the samples are processed for the detection, molecular detection. And follow, of course, as a research part, we subjected some of the samples, not all, some of the samples we are subjected for isolation studies. And these are the details of samples collected and processed for diagnosis of LSD at our center. Different districts, Chittor, Guntur, Kadapa, Nellore districts. Of course, now it is spreading almost all Anantapur and other Vijayanagaram, Srikakulam and Vizag districts. And these are some of the pictures earlier, 2019, these are the pictures we have shown. So including cough, see the cough also having, I think other side is have deep-seated wound. So this is the generalized lesions what we observed during collection of samples. And you see starting from the generalized simple lesion to the complicated deep-seated wounds, what Dr. Manjuna read earlier, he shown his slides also. It varies from mild symptoms to the deep-seated wounds, including cutaneous forms and including the systems also, different systems affecting the, this is the clinical pictures of the samples what we are collected from different forms. And here initially we are subjected for the molecular detection. So using both we are standardized only with conventional PCR, gene-specific primers as well as species primers, targeting P32 genes and G protein, CPRP gene and RPOP gene and anchor repeated genes. So these are the gene-specific and specific genes we are targeted for standardization of molecular detection of Lumpy Skin Disease in our laboratory. And these are the different details of the primers we are used respective genes, and according to the references, according to these references, we are followed the primers, already published primers. We used these things for the diagnosis of the Lumpy Skin Disease at our state diagnostic laboratory. And these are the details of results what we got after processing of molecular detection. And these are the results of the PCR results of the different genus-specific as well as the species-specific, both from the blood samples as well as skin swabs. But from the blood samples, only little number of samples we recovered the positivity of the LSD when compared to the genomic DNA, when compared to the skin scabs. And these are the different species-specific primers what we use to differentiate the LSD from the other sheeppox and goatpox viruses within the genus. So these are the different species-specific primers we are used for standardization. Yeah. So after confirmation of this is for diagnosis, what we have done it. And as a part of PG research program, we attempted some of the isolation of the virus also. So initially, whatever the isolates we got it, we selected only limited number. So 5 to 7 samples were selected as 7 isolates as a PG research program. So initially, whatever the isolates we got it, initially we passaged into the emulated chicken eggs through CAM route. And after these things, we are adapted those into the primary lamb kidney cell cultures. And here, actually, all the isolates, now whatever the isolates we are passaged into the CAM route in emulated chicken eggs. So at the second passage level, on the first day of inoculation, showed embryo CAM coriolantic membrane of the emulated chicken eggs. And only the 3 isolates, of course, we have limited number, we have gone for the isolation, and out of which only 3 isolates show characteristic POC lesions on the CMM route on 5th day of passage level. And after egg passages, again we confirmed the same isolates with the PCR confirmation. So these are the lesions of the CAM route. The first this is the normal CAM route, and so these are the after passages. This is the embryologous lesions what we observed at 5th level of passage in the emulated chicken eggs. And after the embryos, again we have confirmed with specific primers for whether it is virus is LSD or not. And then we adopted this after emulated chicken egg passages. We same the virus isolates. We attempted for further adaptation to the cell cultures. Especially we used primary lamb testicular cell cultures for adaptation of virus into the cell cultures. So initially, at first blind passage level, between 48 to 72 hours of post-infection, so all the whatever the isolates, they show the CPE of light CPE, shrinkage, cell rounding, and aggregation as a part of CPE at first blind passage level. But after 96 hours, complete CPE was observed in those isolates. And these are the primary testicular lamb cell cultures. So these are the normal uninfected cell. And this is the first passage of the 96 hours post inoculation. So this is the first blind passage 24 hours. And this is the 48 hours of post 48 hours of post inoculation, showing the shrinkage of the cells. And this is the first blind process 72 hours. You see the difference, show rounding of cells and cell aggregation. And this is the 72 hours. And after 96 hours, so complete CPE was observed. Whatever the isolates we have passaged, the complete CPE was observed after 96 hours of post inoculation. So this is the complete CPE. This is the isolate. We got it after complete passage. And after after cell culture passage also, again we confirmed the same isolate with the molecular genomic confirming the LSD virus, and this is the picture. And after molecular characterization of these LSD viruses isolates, we are sent for sequencing. This is only partial sequences. We sent for sequencing and confirmed as LSD virus by BLAST analysis. And on multiple nucleotide alignment, the species with the species-specific genes, they show the identity of 99.2 to 99.7 with all the species-specific genes of the LSD, with reference isolates which are available in the gene bank database. So the genome identity is of 99.2 to 99.9%. So confirming our isolates are belonging to LSD virus. On phylogenetic analysis, so whatever the isolates from, especially the isolates recovered from especially skin scabs from the different districts, different outbreak areas of Andhra Pradesh, so formed a separate cluster indicating that there is no strain variation among the recovered isolates of LSD, and these are showing a close relationship with the global reference strains of China and Kenya. So this is the global, this is the phylogenetic analysis. So all these are our isolates, all the isolates forming the separate cluster, and there is very close to each other, and they are very close to the global reference strains of China and Kenya, and have distant relation with our Indian strains like Orissa and West Bengal. So this is the phylogenetic analysis of our isolates on species-specific genes. And this is the another species-specific anchor repeat genes of LSDV showing the same pattern. So separate cluster with a close relation with the global strains of China and Kenya, and a distant relation with the other Indian isolates. So based upon these findings, so the outbreaks are occurring nationwide and also in different districts of Andhra Pradesh, including adjoining states of Andhra Pradesh nowadays. So far there is already, the speakers told that there is no specific vaccine against lumpy skin disease. So however, we are using the goat pox vaccine for control of the disease. And before that, so laboratory, that's what Dr. Manjunath Reddy very well pointed, differential diagnosis is very important. Before we are going for vaccination, we have to confirm the disease because it has differential diagnosis the most of the viral, fungal and other bacterial diseases. So laboratory confirmation is very important, and this laboratory confirmation is standardized in our lab for diagnosis in the field, and also getting the samples from different districts of the Andhra Pradesh and also adjoining districts, adjoining states also. And we confirm the molecular diagnosis for LSD in both the genus as well as species specific with the conventional PCR, and also further confirmed by our virus isolation. So whatever we are standardized procedures are meeting the requirement for a confirmation of the LSD virus. And as a conclusion, so whatever we have done research carried out at our state diagnostic laboratory, SVBU, so the present work revealed that the presence of LSD virus both from the blood as well as skin scabs from different outbreaks of area revealed that the virus is present in our Andhra Pradesh, and it is confirmed by both molecular techniques as well as by cultural isolation. And our molecular characterization studies, of course, we have sent only the little number of samples. With our whatever samples we have sent, molecular characterization studies reveal that there is no strain variation among the LSD isolates what we got so far recovered from the various outbreaks of Andhra Pradesh. However, so the detailed studies required in the state to understand the epidemiology of the disease, and also more number of samples to be analyzed. I think during this pandemic, the 2023, more number of samples should be required for further confirmation of the disease and for strain identification, so that it will help for the planning of vaccine selection of vaccine candidates for to take the action plan for the control of the disease in the state. So this is the as far as our research carried out at LSD work at State Diagnostic Laboratory at Venkateswara Veterinary University. Thank you.
Thank you, Dr. Venkateswara. There is no clear question. But you have concluded that there is a need to understand the second wavelengths, and you have to do the equation part.
Yes, sir.
Now present is a different party of that in 2019. So are you essentially going to take up some projects on this other spot? Our temperatures and also approaching relations to that. So we can assess the variants available in Andhra Pradesh, right? So this
Yeah,
is
sir.
the end of the work. We have taken to this most area, and the PGH to have a major role in any study these areas now. We can look at it. We continue a lot of their demands. Right? So
Yes, sir.
before taking some of the questions, but I'd like to request Vinod Secretary and also Madhura Reddy to react on the validity of the variant we have studied it the phylogenetic studies. They are more related to India and China, really. So now the variants are more of different variants. So what is the validity of this? Based on this, can you go forward or you have to go after study? Now go that.
Sir, I could not hear your voice clearly. Your voice was a bit echoing. Could you repeat your
Yeah,
question?
the study mainly pertains to 2019. So that used mostly the variants pertaining to Kenya and China. They are very unique. They are not very common in other countries.
That's right.
So what is the validity of this particular study? Or we have to go with that study? Or can we, based on the earlier studies, can we go forward?
I mean, I don't have a clear answer to this, but I think a better pertinent question to ask would be, since the 2019 isolate from Ranchi is the precursor for the homologous attenuated vaccine, a much pertinent question to ask would be if we could sequence the present attenuated vaccine to understand more about it. Yes, of course, if we have all the money and resources, we could go back in time and sequence the previous outbreaks in 2019 and 2020 and even 2021. But that would require a bit of money and resources. But if the samples are available, yes, that would also be a useful point to look at, because that will allow to understand how did the virus emerge over and evolve over these years.
Anything that you know, that is a way.
Yes, sir. As Sakaria sir said, if we can compare these three waves, if you have samples, no, so I think you should have present wave also, some of the samples. If they can take up that work of comparing first wave, second wave, and third wave, and that can give you, if you are planning for developing a vaccine of your own for your state, that will give you a clear picture of what is the present virus circulating in AP or Andhra Pradesh. That can be used for further, or whether to use your own strain, which is going to be different from, or it is existing, it is matching. That will answer, sir.
Important question, what are the points of the vaccine? So the current vaccine available, are they homologous or heterologous? Which are the most spoken? What are the
Present heterologous vaccine, can you?
homologous?
Sir, presently, as per the Government of India guidelines, we are using or recommending goatpox vaccine, that is Uttarkashi strain. In case there is not availability, then they may recommend, but in the guidelines, they did not recommend the sheeppox. It is only goatpox which is recommended. And as Tripathi sir was saying, already the vaccine of homologous vaccine, which is commercialized, but it is not yet available in the market, sir. And I think in the due course it is coming soon. As of now, heterologous vaccination is the in the practice, sir. Whether it is in Karnataka or any other state, they are doing with the goatpox.
As far as the immunity or protective concern, nobody takes 100% protection.
Yes.
The cold chain and all.
Yeah, protection, yes, it cannot give you, because it is a heterologous vaccine. It cannot give you 100% protection, so that is sure. But within the available facilities or available logistics in the country, that is giving the, instead of not doing anything, that is giving good productions, sir. More than 80-90% we are observing the productions at the field level. Even we had an interactions with the field veterinarians who are sometimes doing with the sheep pox and goat pox. They themselves will experiment. Field veterinarians are very clever. So they themselves are observing that wherever they are going for the sheep pox vaccination, they are observing or continuing the disease. Whereas, however, on the contrary, if they vaccinate with the goat pox, they are able to see the good results in the science. If even disease develops also, the severity of disease and the spread, everything is less in case of goat pox vaccinated animals.
We have a number of veterinarians participating in the webinar. They can also post their interactions and also observations regarding the potency of factors so that we can share it among the participants. I request the veterinarians in the webinar, participating in the webinar, please post your page. So now I may request Dr. Anand Kumar. Please present your observation because you are the backbone for this webinar. The requesting Dr. to join with that. Dr. Anand Kumar, please give your observation.
Sir, good morning to all. Thank you, sir. Sir, I think my presentation and audio both are clear to you, sir, I think?
Yes, sir.
Yeah, that's fine. Thank you. And first of all, thank you, sir, for giving me an opportunity to speak. And once the first two speakers, Dr. Manjunath and Dr. Vinod Skaria, they have given a beautiful panoramic view about the research component in their own domains, right from the disease pathogenesis epidemiology to the genome sequencing and how can forecast the epidemiology, all these things. So I'm just limiting myself to this particular one, just intricacies and insights. I'm dividing this into three components. One is what happened in 2019 in the coastal districts of Andhra Pradesh, what prompted this laboratory to take up the work. Then the next component, since I am an immunologist by training, what are the things implicated in the pathogenesis? What are the gaps we need to address when we develop the vaccine? That's some sort of insights from my point of view as an immunologist. And third one, since the field veterinarians are participating there, so what they have to do in addition to the available things. So in three parts, I complete this particular presentation. Coming to the first one. So in 2019, we had reports of the Lumpy Skin Disease in the north coastal districts of Andhra Pradesh. So this was the information shared with this laboratory from the Animal Husbandry Department, Government of Andhra Pradesh. So the field veterinarians, they have provided beautiful inputs about the clinical presentation and also the material to our laboratory. Very briefly, I'm showing, I think, certain important photographs which are unique. We could get only those things I'm presenting here. So this is about the cough with the multiple intradermal nodules. All these things are courtesy of Dr. V. Govind. He is presently working as veterinary assistant surgeon in Badangi village in Vijayanagaram district. He took a lot of interest along with his colleagues in 2019. Of course, still right now also he's working and collecting the samples and reporting. And then superficial lymph nodes, how they got enlarged. Of course, these are other lesions. Certain typical lesions also we got something on the muzzle characteristic lesions we got here, ring-like appearance. Of course, these are other things. Very quickly, I'm going through these things. So the characteristic inverted conical zone of necrosis. So we collected samples from almost all these animals in 2019. And of course, edematous legs, necrotic sloughing of all these things were reported. Then typical things here we could find keratitis, Cauda-Eckmann syndrome also we reported from the field veterinarians of the North Coastal Districts of Andhra Pradesh. So what we did when the Animal Husbandry Department in 2019 requested for the diagnosis, we followed the diagnostic protocol of the OA manual, and the PCR test is specific to the oligonucleotide pyramids. We have taken variant attachment protein as per the OE guidelines. We have conducted this one, and Dr. Ramani Pushpa, then working as a professor at this department along with 3 postgraduate students and myself, we together worked on the samples. We adopted this technique and just a diagnosis by PCR. We didn't go forward any other thing. Still, we are skeptical what to do, because for the first time we were hesitating whether to report it or not directly. So these were different samples we collected at the time, and we reported their reactivity, and some samples were positive, some samples were negative. And all these things were reported to the Animal Husbandry Department for further lesser action. After this, we could not take up any other work. Even the samples that time preserved also were not sure of the quality because of the COVID lockdown issue, the preservation, all these things. So this is one component what we have done first time in the Interior College of Veterinary Science of this university at Gannavaram. We have worked on this LSD. Then what is the present scenario? We know this presently goatpox vaccine is widely practiced. Of course, the Lumpy Provac released by ACR is also practiced, but these advantages, limitations of both these vaccines are debated. And of course, there are some other groups they are also working for developing the vaccines. But what actually the important thing I want to stress, Dr. Vinod is there working on the whole genome sequencing and the pathogenesis. Dr. Manjunath is there. And I'm very much concerned. My earlier work with orthopox viruses, it gave me some insights about the molecular mimicry of the pox viruses. So the peculiarity of the pox viruses, so this genome of the pox viruses, they contain certain genes which code for the homologous proteins. Say, for example, certain proteins which can inhibit of complementing, and certain things which modulate the chemokine and cytokine activity, and certain things specifically target the innate immune pathways like toll-like receptors and interferon-induced antiviral resistance. And further, these pox viruses, they encode the chemokine receptor homologs, biologically inactive chemokines, and chemokine binding proteins. The purpose of all these proteins, of course, is to undermine the immune response in a host. So that's helpful for the pathogenesis. And also, this point is to be taken care when any difference or in the genome of this particular lumpy skin disease variants is affecting in such a way or not. This is the most important thing we have to see. The reason is in 2019 we didn't find this type of mortality. So presently we have a lot of mortality, and morbidity is also more. What it is driving? What are the factors that are driving for the pathogenesis and more mortalities? Any of these things, they are driving for more pathogenesis. That means those factors which undermine the immune response are those factors which enhance the pathogenesis. And the answer shall come from the whole genome sequencing and its further analysis. So this is what I request we have to take forward on these aspects. Then only, because we don't find any literature on the lumpy skin disease. We have the literature for about other orthopox viruses about these things. But what's about this type of molecular mimicry by the lumpy skin disease virus in the host, that in different hosts? We don't have any literature right now. But of course, certain proteins, they are homologs of regulator of complement activation. So these things, they will downregulate the immune response or increase the pathogenesis. This is what the most important we have to focus in the coming days. Just even for developing the vaccine, so I think we should not be crazy in the developing vaccines. So developing vaccine for anything doesn't solve the issue unless we take up in a logical way. Say, for example, I came across on the same concept, I came across one particular development of vaccine where the LSDV with the knockout IL-10 gene that gave relatively very good protection. The reason we know the IL-10 which downregulates the interferon-mediated immune response, which is very vital for the pox virus infections. And here, even if we prepare the vaccines, regular activated vaccines, we have to assess what's about the type of cytokines it is producing, because this particular paper, it gave good insights. Whatever the particular knockout with IL-10 gene they produced, that has given a beautiful immunity. So this is the thing. we have to work out, even if we prepare that individual vaccine, we have to characterize it, all these things. Even any other group which takes up this work, in the holistic view, this has to be taken. And the biggest question from the field veterinarians, what we have to do if both are not available or accessible? That means the good box vaccine or the lumpy skin, this lumpy pro vaccine, both are not either available or accessible. So what to do? See, when there is a problem, we have to show the solution. Say certain things, they can't wait for the time. In this one, our own experience, we can say with the pox viruses, this use of ivermectin. That is very latest article in the virus research. This ivermectin, that has beautifully, it is acting in preventing the viral entry and also the viral attachment and also the action of certain functional proteins of the virus. So ivermectin is very widely used in the field. Instead of waiting for other things before the disease gets aggravated, I think our field veterinarians, they can try. These are just highlights of the paper, how effectively in the in vitro cell lines, the ivermectin inhibited the LSDV virus entry or its effect. So this gives us some clear indication, and there is nothing wrong in using the ivermectin also when you don't have any other sources. And in addition, these are all the vast literature, right, from 2009 to 2022, how the ivermectin acted on different viruses and inhibited its viral entry, multiplication, and secretion of different functional proteins. All these are different references where you can find the action of ivermectin in the SARS-CoV-2, in the yellow fever, in the West Nile virus, Newcastle disease virus, influenza virus, pors and respiratory disease virus, equine viruses, so many things, how it inhibited the entry, and also the targets of inflammation, also how this ivermectin targeted, right from acting on the toll-like receptors, NFKB pathways, LPS-induced stimulation, and all other pathways, and also the activated kinases, also how it controlled the inflammation. So this is the good example. If you don't have any other sources, all these things are summarized in this particular article. JD and Degani Mubarki, they summarized the ivermectin action on all the viruses. So this is a beautiful one, and we can go ahead if you don't have any other sources. And what's about the natural products of antiviral and immunomodulatory activities? We have some good experience with the research project funded by National Medicinal Plant Board. We tried with some anti-medicinal plants. In fact, that expertise helped us to for the COVID research also there. It worked well. And when it comes to these viruses, we have the andrographlides, the very common plant Andrographis paniculata, Telugulo Nelavemandamu. So we have the established evidence about the inhibition of viral entry stage, viral gene replication, formation of functional proteins, treating these andrographlide plant extracts. Even in the SARS-CoV-2 also it is established. And further, the inflammatory cytokines also, they can be down-regulated. And I'm surprised to see during my interaction with all the veterinarians working in different districts in this LSD scenario, many field veterinarians in the North Coastal districts, they have told the farmers, they have already using this plant extract of this andrographlide, Andrographis paniculata to the cattle as a precautionary measure. Even they're trying also this one. Surprisingly, of course, we can say this is a complementary therapy unless we have full-phase studies, but it's working. So how it works, all these things, we have evidence beautifully described in this paper, antiviral activities and andrographlides, derivatives, mechanism of action, and delivery systems. And these are different plant components which have the anti-inflammatory properties and antiviral properties of this paniculata. To just give few examples how in different infections, this is how in the influenza virus this particular plant worked, and how it worked in the HIV infections, down regulating the chemokine receptors here, and how in that Epstein-Barr infection, different pathways of the lytic cycle, they are interacted and acted as antiviral activity. So these are all the things, the way forward about the immune responses, or what we say the molecular mimicry, and the immediate need when you don't find other alternatives of the vaccine or other things. So with this one, I conclude. Thank you, sir. The thing is open. Thank you.
Thank you, Dr. Kumar, for writing the literature. Definitely. So many of these have done I think many products in the This is, I think they are working well with the veterinary doctors in the field. Definitely, considering radiation. Since they are applying like a topical application, we plan to do it working at again the same and
Yes, so some of them they are going for the topical application, and some of them they are feeding it as a paste also. In Vijayanagram district and Srikakulam district, when I interacted with them through the veterinarians, they are using this particular. Of course, they may not be knowing the molecular evidence, all these things. Maybe the traditional veterinary practices they may be doing, and we have a lot of scope for the research also on these areas, along with developing the vaccine and also other things.
there is new veterinary doctors on the board and the participating in webinar. When you share the experience of the SOP for the cheat that you are successfully achieved, and thinking you get to on any of the scientific that you photos there. So they have posted the economic impact of the LFC with regard to the change from the posting. You see, post the important line of treatment. Your especially the really practice that really the basis to be consistent. Thank you, Dr. Anand. And
Thank you.
yeah, before getting to the next speaker, I'd like to post a bit on the that we know that also the so there are reports of affecting the human brain. We have seen many literature available, so that's also affecting the human brain. Now, I think I learned from Dr. Manjunath that it is not the process. But there are reports of this. How is going to be
Anand sir, you're going to answer or can I answer, sir?
Yeah, please. Because I'm keen. These are my purpose. What's going to be the future of engagement?
Yes, sir. Your question is definitely relevant. The two papers that are one paper, it has came out from Brazil, is he did not associate even the disease with the clinical signs. Sir, in that finally he said that it is herpes virus infection. In that on the lesion, he could find the genome. He could not neither isolate the virus nor as on date, nor this World Animal Health Organization or OIE, they did not consider these findings as suggestive of zoonotic in nature, and neither it has been proved with postulates where we have to repeat the disease, we have to isolate the virus from these lesions. Accidentally, if, for example, tomorrow today, if I am going for an outbreak, if I touch the animal without wearing proper PPE, and I had a wound, if I go to an hospital, if they test, definitely the virus will be there on my skin. So that does not mean that the disease or the person got infected. Infection is different than the detection. So with that, that's how the people still say it is a non-zoonotic disease, sir. I hope I have answered this. And recently, in the science, since last 3 years, we are doing a lot of field investigations. Ourselves are examples in the sense. I hope you may not have received such kind of complaints from the field also. Anybody developing any clinical science, sir?
And one more
yes.
When like a secondary infection is
Secondary infection with the virus or bacteria, sir?
But this kind of things like nodules are there, open so any secondary infection are such things are infection. Such problems definitely complicated. Real.
No, so in animals, you are talking about secondary bacterial infection. Yeah, in the animals, definitely there are secondary bacterial infections of the wounds, depending on the kind of antibiotic they are using. And sometimes, the farmers have the practice of giving bath to these dairy animals every day. So during that rubbing process, you might have observed they may use some scrub or some plastic brush. During that process, they do cause a lot of damage to the skin, and secondary bacterial infections are quite high, sir, in case of these LSD affected animals. We are trying to isolate even, there are some lesions of in the lungs of the affected or dead animals. We could able to see some of the HS kind of post-mortem lesions, but isolation we are unable to do it, because the moment you inject antibiotic, the HS organism is going to be killed. But histopathologically, or the post-mortem signs are suggesting there are definitely secondary bacterial infections, whether it is a systematic or on the skin dermatitis form, sir.
Okay. You confirm that the milk is safe for consumption?
Yes, sir,
Of
yes.
as well as the liver.
Yes. The milk is safe for consumption, and it should be boiled, and as usual, to avoid any other zoonotic diseases like brucella or TB. So it is safe for consumption until unless the cow is suffering from mastitis. The differentiation here is, and clinically positive animal with LSD, it may not be a positive for mastitis. If the mastitis is there, the mastitis is caused by some secondary bacterial infection. It's not due to the LSD. However, if the udder is clinically healthy, the cow is infected with LSD, the virus is excreted during the last stages or the terminal stages in the milk. The milk, if you boil and drink, whether it is for human consumption, or we advise even for this indigenous cows, where do we let the cows to suckle the animal, instead of allowing the calves to suckle, because the teeth are getting already lesions or damaged, so there is high probability of causing the mastitis. Hence, the milk can be, the animal can be milked, and even you can feed it to the cows, the boiled milk, because the milk is one of the source for the cows, the infection.
Okay. Thank you very much. And now, speaker, may I request Dr. Lakshmi Kavda, Professor in microbiology. I think we along with other scientists to first to see what the first to do some research and like there's a information there. May I request Dr. Lakshmi Kavda to please present briefly the research.
Sir, thank you, sir. I think I am audible to all of you, and my presentation is visible.
Just go ahead with that.
Yes, sir. Thank you, sir, and good morning to everyone. And I take this opportunity to express my sincere gratitude to our Honorable Vice Chancellor, Sir, Dr. V. Padmanabh Reddy Garu, and Honorable Dean, Sir, Dr. Sajjan Reddy Garu, for giving this opportunity. And also, I take this opportunity to greet Honorable DDG, Animal Sciences ICR, New Delhi, on this forum, and also other scientists and dignitaries and dear participants. So, all the speakers they have given a very good insights about the lumpy skin disease, and in this context I would like to explain, and I would like to explain the work done by us, along with some of the perplexing points that are there that needs to be addressed for the field veterinarians. So this is the overview of my presentation: etiology, epidemiology, pathogenesis, clinical science, diagnosis, treatment and control, and roadmap. So what to do after going through this. So as already explained about the etiology, this is a double-stranded DNA virus having 151 kilobase genome size, and it is having 156 genes, with a central core having the conserved type of genes, whereas the terminal genes having variations with and they are coding for host specificity as well as the replication, etc. And when it comes to the cross protection, etc., as this virus shares genus with goatpox and sheeppox virus, and this is the structure of the virus with a clear-cut dumbbell-shaped nucleic acid, as well as two lateral bodies with one outer membrane and external membrane. So these lateral bodies and the dumbbell-shaped structures are the one which, through this, we can pick up the virus in electron microscopy very easily. And when it comes to the epidemiology, this is the one which has started in the Zambia and has distributed to several parts. The first outside distribution of this virus from African continent occurred in 1989 to Israel. And later on, it has spread to different parts like Jordan, Iran, Saudi Arabia. And in 2019, observed in Russia, China, Bangladesh, India. And 2022, the several countries surrounding India has observed this outbreak. So this is the FAO given maps regarding the disease outbreaks from 1989. The first map explains 1989 to 2018. Second one, 89 to 2019. So in 2019, outbreaks occurred in India. And at 2022, there is a wide distribution of this particular disease. Then in 2021, major number of events has happened. And cattle is the worst hit compared to buffaloes. And Asia is the worst hit by this virus in the recent past. And as per FAO, 20 events were observed in India. And this is the one what we did. We have collected samples from 4 different regions, Chittoor, Nellore, Prakasam, as well as Krishna district, and observed the presence of virus based on the clinical as well as the molecular diagnosis. And this is the one which spreads very fastly from one area to another area. So the transmission of the disease, infected animals, direct and indirect transmission, vector transmission, direct through discharges, intrauterine transmission, and indirect through fomites, vector transmission. And there are several vectors that play an important role. And these vectors are the one which are required for the long distance transmission. Even the outside spread of Africa is also because of the long distance transmission through the animal carrying trucks, especially by the vectors. So from the affected area, infected animal and infected vectors are the sources to infect LSD free zones. So the different risk factors. So epidemiology is a very, very important component to understand the disease process and also to understand the virus dynamics. The different risk factors that play an important role in the occurrence of this disease. One is the host. So among the host, cattle, Bos taurus is more susceptible. Buffaloes are also susceptible. And the other hosts are giraffe, impala, Arabian oryx, springbok. Crossbreds, there is a in some cases, they are saying crossbreds are more susceptible. In some cases, they are equally susceptible. And all ages are susceptible. Among them, young ones are highly susceptible. Lactating cows are more susceptible and weak ones are more susceptible. And coming to the environmental risk factors, hot and humid climatic conditions and more than 1000 millimeter rainfall, where the areas are having more than 1000 millimeter. In India, average rainfall is 1180. And fenced farming, communal water sources, and vector population. There are several vectors that transmit, especially the biting midges. Then coming to the risk factors related to the pathogen. The pathogen secretes in different secretions like saliva, lacrimal discharges, nasal, blood, milk, and semen. The LSDV endures for 33 days in skin nodules, scorched scabs for 35 days, 18 days in dried skin, and gets deactivated at 55 degrees centigrade for 2 hours and 65 degrees centigrade for 30 minutes, and survives for 6 months on fomites, and it survives in vector not more than 4 to 7 days. So in vectors, the virus gets transmitted through mechanical means. But there is one report stating that a ovarian transmission has occurred in rhipicephalus ticks, which needs to be further elucidated. Economic impact, there are direct and indirect losses. The estimated losses are about $6.43 US in local cattle and $58 US in HF and crossbred, as per Gary et al. 2011 by this time. this becomes more costly. So the direct losses are loss in milk production, abortions and fertility losses, emaciation, damaged skin, loss of productivity, indirect losses in terms of trade restrictions, supportive treatment, disinfection of farms, farm premises, vaccinations, vaccination production costs, and management of laboratories, etc. Then this is the virus which replicates especially in the epidermal tissues, and later on it replicates in the endothelial macrophages, endothelial cells and fibroblasts. So this is the area where the virus is causing majority of the intravascular damage, which leads to several complications. And also in the fibroblast pericytes, and 4 to 7 days cutaneous nodules erupt, and 7 to 19 days lymphadenopathy. And because of the lymphatic drainage, blockage, etc., some of the animals are showing edema, all these things. Then at the 40 days post fever, virus found in semen. So this is a very important point which needs to be stressed, especially in the breeding stations. Then coming to the clinical signs, fever more than 41 degrees centigrade, elevated firm demarcated skin nodules, anorexia, nasal discharges, drop in milk production, ulcerated and necrotic nodules, enlargement of limbs and lameness, secondary bacterial complications, abortions, temporary or permanent infertility in male. The morbidity goes up to even 100% in some areas, and mortality 1 to 5% goes up to 20%. So these are the some of the clinical signs what we observed. So this is moderate cough.
Yeah, please present more on your research.
Okay, sir. Sure, sir. So this is a cow and calf that gets infected. The calf get infection from the mother, and these are the some of the clinical signs observed, and lymphangitis in case of Ebola, and these are the animals that were affected. Then diagnosis, among the different samples, skin nodules gave a very good result in the diagnosis of the disease. There are several diagnostic approaches, clinical signs and isolation, as well as the molecular diagnosis and virus neutralization. As per OAE, confirmation of the clinical signs can be done by virus isolation, PCR electron microscopy, and antibody response by virus neutralization and IFAT, that is indirect fluorescent antibody technique. This is the differential diagnosis of skin lesions. It has to be differentiated from several diseases like pseudo-lumpy skin disease, etc. And this is the one what we did. We utilized ORF 117 primers for the specific identification of LSDV, as well as we utilized reverse polymerase 30 gene for the amplification of this virus. So confirmation was done basing on these two PCRs. Then molecular characterization. So this molecular characterization is very crucial to understand the entry of the pathogen into the area, as well as to understand any genomic changes that are taking place in the virus. RPO 30 gene sequencing and phylogeny has given an insights about the path of entry of virus to Andhra Pradesh. That's from Orissa, Bangladesh, Russia, Kazakhstan, and Iraq, and later on to the South Africa, basing on the phylogenetic percentage. And this is the article published by us on Dr. K. Lakshmi Kavita, Sridevi, and Dr. Rajesh. They published an article on clinical molecular diagnosis and characterization of bovine lumpy skin disease. And this is the ML trees that has given phylogeny with the other circulating viruses, especially in India as well as surrounding India. Then these are the some of the treatment protocols that are being used, giving a secondary to arrest secondary bacterial complications is very much crucial. And NSAIDs, especially flunixin meglumine, is the one which gives a good result. Fluid and electrolyte therapy, and Veterinary formulations given by NDDB in the YouTube channel in different languages, and homeopathic formulations are also there. Now, a very important aspect is the vaccines. So what is happening, and there are some perplexing points or baffling questions given by the field veterinarians, especially regarding the vaccines. So as of now, globally, there are several vaccines available, and majority of them are based on the neethling strain, and only the Kenya Veterinary Vaccine Production Institute, it has used Lumpy Vax, which is mainly based on the Kenyan sheeppox and goatpox virus. whereas the other institutes, they utilized the sheeppox vaccine, whereas Russia used sheeppox to control the lumpy skin disease virus. What we are taking in India? Attenuated Uttarkashi goatpox vaccine is used and immunity for 1 year, and a lumpy Provac, a homologous vaccine, is under approval for emergency use. Whereas Bangladesh, it has procured Lumpyvax from MSD Animal Health, and immunity from homologous vaccines is about 2 years with Kenyan strains and 3 years with South African strains. And this is the Lumpy Provac, which is released in the August 2022, which is a homologous vaccine that has been attenuated. Then what is this homologous vaccines? So the South African neethling strain, it has been passaged for about 81 times. Madagascar LSD strain passaged for about 100 times to be get attenuated. The culprit here is the KSGP O240 and O180 strains. So nowadays, everyone is having a doubt about the vaccine recombinants and where from these vaccine recombinants appeared. So here the KSGP O240 and O180 strains, as per literature says, this is the strain which actually it is a lumpy skin disease virus, and it has been less attenuated for about 5 to 30 times in the laboratory. And also this vaccine batches contain different viruses, not only the KSGP, it also contains Sudan goatpox and other virus, which made the exchange of genetic material has happened in the laboratory, especially during the vaccine production, but it hasn't happened in the co-infected animals. So now majority of the field veterinarians, they are expressing their doubts that whether any vaccine recombinants are going to cause, going to occur or something else like that. But this is what happened in the 2016, and it led to the 2019 outbreak in Russia. Then heterologous vaccines, especially the goatpox. Now India is using goatpox vaccine. As per literature, goatpox vaccine 20 times attenuation is sufficient to give a good protection to the cattle. But our Uttarkashi strain got 60 times passage for its attenuation and is highly safe for usage. And the heterologous vaccines has given 70 to 80% efficacy. And heterologous sheeppox vaccines, they have given partial cross protection or incomplete protection was observed by several researchers. Then how to administer inactivate? One of the important points that needs to be considered during the administration of capripox vaccines is these viruses are highly susceptible to the sunlight. So they get inactivated by the sunlight. So every time the proper practices, standard operating practices should be practiced during the time of vaccination, keeping the vial at cold temperatures. And all currently used homologous and heterologous vaccines can be given by subcutaneous route. And the minimum dose of neethling vaccines are TCID 53.5, and the minimum dose of goatpox vaccine is TCID 53. Now, what about the variants observed in the recent outbreak? Dr. Vinod Skaria, he explained very beautifully about the latest variants. And one more small point I would like to add in this is the variants observed are the single nucleotide variations or indels. They are mainly observed in the immunomodulation, virulence, and host range, whereas the difference between the vaccine variant and the attenuated neethling variant, that is attenuated neethling variant with that of the virulent virus is in the hypothetical proteins LSO 37, 037, 114, and 156. So this could give a better insights to understand something is happening, or some changes are happening in the virus while it is acclimatizing to the new geographical conditions, or it is spreading in the new host, or else the host factors are responsible for this type of severity needs to be studied. And the studies on the inactivated and recombinant vaccines should be undertaken. Then what should be our roadmap in controlling this particular scenario? Restrict the movement of sick animals, regular observation and testing, which is very, very crucial. And I take this opportunity to request our honorable authorities to give us spare some amount of samples to work on it, and also to go for phylogenetic studies, and also to come up with rapid diagnostics and epidemiological forecast at least. Quarantine is very, very required. decontamination of the infected premises. So farmers can use 1% formalin as decontaminant, 2% phenol, and 2 to 3% sodium hypochlorite, which are easily available. And virus is susceptible to sunlight and lipid solvents, and vector control is very crucial. And in severe areas, dividing the area into different zones, what we did for COVID-19 to control the spread of this disease: frontier vaccination, that is vaccination at the borders, and ring vaccination for 20 kilometers, closing of borders and avoid mass migration, ban on shanties, prohibition on movement of animals from infected to uninfected areas. So if we could able to plan the arrest the movement of animals from the epicenter, so epicenter, control area, and outside area, we could able to divide the actual outbreak area. We could able to control the spread of the disease. As this is a vector-borne infection, vector control is very crucial. Using mosquito nets, burn neem leaves, spraying acaricides, insecticides, clearing vegetation, and one of the important plausible way of transmission is the iatrogenic transmission. Always use a sterile syringe needle for vaccinating or any kind of injections that is taken to the animals. And what should be undertaken? Extensive studies on the epidemiology is very, very crucial to understand the disease spread and virus dynamics, exploring the possibilities of rapid diagnostics and development of DIVA vaccines and diagnostics. And thank you for giving me this opportunity.
Very good presentation. So we have given enough information on overall overview of the prevention as well as for the But at this time,
Dr. Kavita, Dr. Sarjan, I have a
yes, sir, please.
clarification. Dr. Kavita, for the information of all of you, that the ICR developed vaccine, Provac India other vaccine has already been commercialized, and now Viavet has taken this vaccine, and 2 more companies have already lined up for. They have also submitted fee, and they are also taking the vaccine. So I think very soon homologous vaccine will come up, and this vaccine has been able to neutralize. By it has got it has been tested on the sera available currently, meaning that since the virus is from 2019 Ranchi strain, and today's virus, so this vaccine has been able to neutralize, produce good protective antibodies as well. Though in all capillary viruses, it is CMI also that plays the role. But we have seen that current variation, some genetic variation that has occurred, some mutation, that is not a major change. And this vaccine is able to protect all strains. That is all the information of all of you. So very soon, I think this vaccine will come in the market.
Sure.
Mr. for the information, separately. On the forward from the society, looking for that kind of commercial. So the vaccines are available. We can deal with the thank you for the information. Sometimes we take the case and effectively all the farming chemicals we have there. Department of Animal Husbandry, Another important point is that the 3 scientists are here, and now other scientists planning the board. So I request you to present a small poster which shows the SOP of prevention of the disease, because there is a lot of prevention of the vaccination and also cold training. We have to do some SOP as well as the therapeutic along with the medical people. So I request all the scientists of HMU, please prepare a SOP, standard operation practice prevent symptoms of the disease, and we will release to the veterinary so that we will follow it. So please follow make such approach. So we have got done some diagnostic check, and by taking the information from these scientists, we can take it forward for the extension department. Okay.
Yes, sir.
Coming to the few questions we have to Dr. Venkateswara and also Dr. Manjana. We are available. Please I'd like to know the species-specific immunity. Why it is not much prevalent in buffalo? Is it something related to the heat or the immunity levels of the inherent immunity levels of the species? And also, why the difference between the cross stress and the indigenous status? We have seen the report that the indigenous animals getting less affected than the cross stress and exotic. Other important question is what are the important predominant epitopes crucial for immunity in animals? May I refer to Dr. Manjunath and Dr. Vinod to react. Dr. Manjunath, Dr. Vinod, over there.
Yes, sir.
Yeah, you heard my question.
Yeah, first question regarding buffaloes, why they are not suffering as good as that of cattle, right?
Yeah.
Yeah, still, as for our observations, what we have seen is a very mild form of disease, and people are more concentrating, or because the clinical signs are more pronounced in the case of cattle, so we are concentrating on them. One, definitely the disease expression pattern is very mild in case of buffaloes, and this may be a species resistance that is yet to be confirmed or studied, and we can expect in future maybe again it is an hypothetical this one, the strain may get adapted the way it is got adapted in 2019 to cattle, the way it is from 2020-23 what we are finding highly virulent or form of disease. In the same way, in buffaloes also, we may expect such kind of epidemics or the severity change of the disease. One more thing is because India is endemic to the buffalo pox, or buffalo pox has been already reported in India. Again, and that might be the one of the other factor where they have a certain kind of immune or like COVID, no, we in India they said that Mycobacterium tuberculosis vaccine we are using, hence it is giving some kind of cross talk with the COVID. In similar way, buffalo pox is there. Whether that is also giving some kind of protection for them. These are some of my this ones, but yes, there are definitely studies required to answer these questions. Regarding the epitopes, I think Anand sir may answer better, sir.
Yeah. Dr. Vinod, what are the predominant epitopes for immunity?
Yes, so thanks for the question. So in terms of the poxviruses, though they are very conserved in their amino acid sequences, to be frank, not much of research has been done on lumpy skin disease, or for that matter, even goat pox or sheep pox vaccine epitopes. Almost a majority of, when I say majority, almost 99% of all epitopes that we know today in pox viruses are thanks to researchers who have worked on the vaccinia and the variola viruses, which are, of course, a different lineage of pox viruses altogether. But having said that, there seems to be some level of cross protection between the pox viruses. But frankly speaking, we do not know much about the lumpy skin disease epitopes, nor the goat pox or the sheep pox epitope at this moment.
Good. Thank you very much. Very important. Now it's time to the first Rahul Chaudhary from the VA. Finally, the concluding word and then very first Rahul Chaudhary, VA.
Thank you, Professor Reddy. On behalf of Unnat Bharat Abhiyan, IIT Delhi, I would like to thank all the speakers for their sharing knowledge and the precautions and other tips and the guidelines to stop this lumpy disease. My sincere thanks to Dr. B. N. Tripathi sir, Dr. P. Baduna Reddy sir, Professor V. K. Vijay, and especially Dr. K. Srijan Reddy. I also thank you, our YouTube audience, for the active participation. I'm sure they will use this knowledge whenever they require, they feel that that should be transferred in the village, they will do it. My sincere thanks to Professor Reddy, because sir has agreed in a very short notice to organize this webinar, and he has taken care of all the things like inviting speakers and arranging them. So I personally thank you, sir, and thank you all. That's all from my side, sir.
Thank you.
Thank
Thank
you,
you very
sir.
much, all the participants. Please follow.
To all, and happy Dasara to all of you. Thank you for inviting, sir.